everyone, my name is Cathy and I'm a prion protein scientist. From your assignment this week, you learned a little bit about prion diseases, especially your assigned disease of interest. We'll be talking more about these different diseases in our next class. In this video, you'll learn more about the prion protein itself. Specifically, how do prion proteins cause disease in humans or in animals? What exactly is going wrong? In order to understand what goes wrong, we're going to need to think back to earlier in the course when you learned about proteins. You might recall that amino acids are the building blocks of proteins. Individual amino acids get linked together into a long chain called a polypeptide. To make this more visual, we'll use some craft supplies. Here is our model of a polypeptide, with individual amino acids, represented by the pink balls, linked together in a chain by covalent bonds. Here, all of the amino acids are the same, but generally, you'll have a sequence of 20 different types of amino acids. Different proteins have different sequences, which are encoded by the sequence of base pairs in the gene that codes for your protein. Now, our polypeptide chain doesn't just stay unfolded like a wet noodle. It gets folded into different three-dimensional structures. The structure of the properly folded protein is determined by the sequence of amino acids, and the proper structure is critical for the protein to carry out its function, whether it's an enzyme in the cell or a protein involved in making the cytoskeleton structure. What causes the protein to fold up, though? Well, it's things like hydrogen bonds, thinking back to your introductory chemistry courses. So those are non-covalent interactions between different atoms in the molecule. Another big one is the fact that oil and other fatty substances don't like water. So the amino acids that are less like water tend to cluster up in the inside of the protein. So they cluster up in the middle, hiding away from all the water that's floating around on the outside. Now, what does this look like? Proteins are very small, so we can't really see this directly. But scientists have methods by which we can figure out the three-dimensional structure of the protein indirectly. You might remember the story of DNA, when Rosalind Franklin passed X-rays through DNA to get an image that then was a critical step in determining that the structure of DNA was a double helix. So similarly, scientists have found out that the polypeptide chain, which we call a backbone, can also fold up into special structures. Not a double helix this time, but we can make a helix. Now I'm using beads and pipe cleaners to show you what's called an alpha helix. So here we have, on the black pipe cleaner representing the backbone, we have different beads. So each bead is representing an amino acid. So the beads of the same color represent the atoms in an individual amino acid. So in the blue, for example, those would be one amino acid, and here are some beads representing it. There's multiple atoms inside it. So you can see that the backbone here spirals around. Another way that the protein can fold is called a beta sheet. The backbone forms a zigzag pleat, and then the pleat can interact with other parallel parts of the chain. So here if we have another chain, you can have interactions between another beta pleat to form a beta sheet, and basically a whole sheet of interactions. Then, these individual regions of helix and sheet, as well as some other regions that aren't as well folded, so they might be less structured, maybe a turn, or a part that's actually mostly unstructured and wet noodly, they all come together to form the three-dimensional structure of the protein.